right, continuing on with 11.2, Algebra 2, 11.2. This is part B for 11.2. Our learning target is I can identify a sampling method. So how these samples are chosen, these are our sampling methods. A simple random sample would just be each member of the population is equally likely to be chosen, and each possible sample of the size you want is equally likely to be chosen. So that would give the most valid results. But there are 5 types of sampling methods, and we're going to go over all 5 of those now. So the first 3 we're going to talk about is less likely for bias, and the last 2 would be a higher risk of bias. Remember, bias being the misrepresentation of the population. So, all right, first 3 sampling methods, the better of the 5 because they are less likely to have bias. So the first one we have is a stratified sampling. Says when a population is divided into groups with similar characteristics and a sample is chosen randomly from each group. So I like to say that this is a sample of a sample. You're taking a sample and then you're pulling from that sample to create your groups there. The next one is called cluster sampling. Cluster sampling is when a population is divided into convenient clusters and entire clusters are chosen as a random sample. So this kind of sounds the same as stratified sampling, except for what's different with cluster sampling is that the entire cluster is chosen. Where with stratified sampling, you're just pulling some of that sample instead of the entire sample. And then our third one with the less likely to have bias is the systematic sampling. Start with one member chosen at random, and then you follow a rule or a pattern. Let me give you an example. Say you pick one person at random and then you choose every third person. So a group of people are walking down the hallway and I choose every third person to ask my question or conduct my study. All right, the last 2 with a higher risk of bias are the convenient sampling and the self-selected sampling. So those are our last 2 of the 5. Convenient sampling is where you choose people that are close to you or easy to get to. And then self-selected is sample made up completely of people that volunteer to be in your study. So volunteers would be the key word there. So again, I have just kind of highlighted what's important for my success criteria. Stratified sampling, we're calling that the sample of a sample. Cluster sampling is the entire clusters are chosen. Systematic sampling is the rule or the pattern. Convenience is when you're choosing people that are close to you or easy to get to. And then finally, self-selected is your group of volunteers. All right, moving on. Some practice with this. Go ahead, pause the video and answer these questions. All right, let's go over the answers. For A here, this is going to be systematic. You're looking for that rule of every 5th student. So there's your pattern. That makes it systematic. B is self-selected. We're looking at volunteers. Only the customers chose to do this. So they're volunteering. So that's self-selected. And then C, They're only going around City Hall where they're close to there. So that is convenience. C is convenience. All right, going on. Example 3 here. A is systematic, not likely to be biased. And then we have B, that is a cluster. The population is divided into grade levels, and then entire clusters are selected. And then lastly, C is self-selected because the subjects are volunteering there. And then, yes, that would be a bias because, remember, that's one of the two that are highly likely to be biased, the self-selected and the convenience. So, yes, that is likely to be biased there. Okay, so that completes that learning target. We're going to go on to our second learning target for this lesson. Learning target, I can design an experiment. So let's just review. We've talked about this before in the last video, but experimental group is the group that receives the treatment. Control group does not receive the treatment or gets a placebo. And this is new. We haven't talked about this, but you might have heard of this before. A placebo is like a fake treatment. So for example, if you were doing a study about medicine and you want to make your two groups and you give your experimental group the medicine, but your control group, you give just like a sugar pill, a pill that wouldn't affect them in any great way. That would be called a placebo. So this group thinks they are getting the treatment, but in actuality, they are not. That would be a placebo. All right, success criteria for designing an experiment. First, you have to assign your experimental group, which group is going to get the treatment, and then you're going to assign your control group, which group will not receive the treatment, or maybe they will receive a placebo. And then lastly, you're going to compare the results of both of those groups, and in comparing those results, you will be able to make your conclusion. All right, go ahead, pause the video and try these two examples. Okay, example 4 here. So this is an example of an experiment here. So let's look at those 3 key elements. So we have the control group that does not get the drug, and then we have the experiment who does receive the drug. And then lastly, you are going to compare your results. So finding the difference between the two groups, comparing those two groups. So those are your 3 parts to your experiment. Okay, down here, they're asking you to test whether drinking coffee improves memory. So using your success criteria, you would assign an experimental group. That would be the group that receives the coffee. And then you would assign your control group. That group would not receive coffee. They would not drink coffee. And then finally, you would compare the results of the two, the coffee drinkers and the non-coffee drinkers, and see how memory was affected. All right, that's all I have for you for this lesson. So this concludes chapter 11.2.